An action potential is defined as a rapid change in the cell membrane potential that is generated only in excitable cells such as neurons and muscle cells. In this process, the resting potential of the cell membrane becomes temporarily positive and immediately returns to its initial state. The action potential is caused by the ions exchanges between the two sides of the cell membrane, which are done by the voltage-gated sodium channels as well as the voltage-gated potassium channels. These channels are sensitive to changes in cell membrane voltage and are different from the leak channels involved in the generation of the resting membrane potential. Voltage-gated sodium channels have two gates, the activation gate and the inactivation gate. Voltage-gated potassium channels have only one gate. When a cell membrane is in the resting state, the potassium channels gate and the sodium channels activation gate are closed, while the inactivation gate is open. The presence of a stimulus changes the potential of the cell membrane. When the cell membrane voltage becomes positive enough, the activation gate opens. Sodium ions diffuse from out of the cell to inside and make the cell membrane depolarized. After a very short time, the inactivation gate closes and sodium ions can no longer enter the cell. At the same time, the potassium channels gate opens. Potassium ions leave the cell and make the cell membrane repolarized. Towards the end of repolarization, the cell membrane potential is still slightly different from the resting membrane potential due to the outflow of potassium ions. Therefore, the sodium-potassium pump becomes activated and moves sodium ions from inside the cell to outside and potassium ions from outside the cell to inside. Now the cell membrane reaches the resting state. The steps of an action potential are shown on the graph as follows. In the resting state, the cell membrane is polarized and its potential is about -70 millivolts. The stimulus increases the cell membrane potential, but its amount may not be sufficient to generate an action potential. The cell membrane potential reaches the threshold level when its potential becomes positive enough. The threshold potential value is about -50 to -55 millivolts. At the threshold voltage, sodium channels open and sodium ions diffuse from out of the cell to inside and make the cell membrane depolarized. At this time, the cell membrane is highly permeable to sodium ions. At the peak or overshoot phase, sodium channels close and the delayed rectifier potassium channels open. The outflow of potassium ions initiates the repolarization of the cell membrane. However, the efflux of many potassium ions makes the cell membrane hyperpolarized before reaching the resting state. By the operation of the sodium-potassium pump, the cell membrane voltage reaches the resting potential. After the action potential is generated, the excitable cells can no longer produce an action potential for a period. This period is called the refractory period. In the absolute refractory period, no action potential can be generated due to the inactivity of voltage-gated sodium channels. In the relative refractory period, the cell membrane is hyperpolarized, and an action potential can be generated if there is a very strong stimulus. To generate an action potential, the stimulus must have enough energy. The threshold stimuli are strong enough to generate an action potential. Suprathreshold stimuli are more powerful than threshold stimuli and can generate an action potential. Some stimuli cannot generate an action potential. They are called subthreshold stimuli. Therefore, the action potential behaves according to the all or none law. That means an action potential is generated or not generated at all. Threshold and suprathreshold stimuli generate an action potential, while subthreshold stimuli do not generate an action potential in excitable cell membranes.